so what is ecotoxicology? So what we're trying to do is determine toxicity thresholds that are associated with chemical contaminants, so environmental pollution. And we want to determine what levels cause mortality. That's a common regulatory endpoint, so understand that. But then also develop sublethal indicators of contaminant exposure and stress. And we also want to develop sensitive analytical methods so that we can go out into the environment, collect water or sediment or organism tissues and be able to measure those chemicals in them and be able to then compare the results present in the field to those levels that caused harm in the laboratory studies. So we're looking at priority pollutants. That means, what's common of concern right now. And we'll talk about some of those in a little bit, as well as contaminants that are just really persistent and have been maybe banned for years, but they're still found in the environment. And then we want to combine some chemicals together so that we can say, okay, here's what they look like individually. Here's what effects we see when we put them together in mixtures. And then add in multiple stressors. So climate factors play a role in how chemical contamination causes toxicity. So we want to do some of those multi-stressor exposures. And then ultimately, these data will be used to improve risk assessment, both for animal and human health. So we also try to look at where the chemicals are coming from, how they're transported in the environment, what different compartments they will move to, and then how will they break down. So that's chemical transport and fate. And then we support the national and regional monitoring efforts where we're trying to assist the collection of the samples and then being able to bring those back and analyze for a bunch of different chemicals. So we do a lot of work for NOAA's mandate for oil spill response and restoration, but it's not all oil work. There's a number of different chemicals that we look at. That's right, but this is device 5.4. Okay, so what are some of the capabilities that we have for doing this? So what's in our toolbox? So we conduct acute and chronic toxicity tests. So we're setting up levels of chemicals to expose the animals to in the laboratory using both water and sediment exposures. These can be static or they can have renewed water and chemical treatment or can be flowed through a continuous exposure. And that matters because there's different ways that you'll experience those exposures in the field, and we want to mimic those in the laboratory. And then we're also looking at cellular and molecular biomarkers. So like I said, mortality is an important endpoint, but then also can we detect stress earlier using cellular and molecular tools. Our analytical chemistry group has the capability to measure inorganic and organic contaminants in water, sediment, and tissues. We do studies to look at the rate at which those chemicals are taken up, and then you move the animals to clean water and measure the time it takes for them to degrade those chemicals back out. That gives that persistence and the potential for things to bioaccumulate and work their way up the food web. So that's why that's important. So like I said, it can be things that have been around, legacy contaminants like DDT that are really persistent, or things that are called chemicals of emerging concern. So these would be more things like PFOS, microplastics, things that are coming to study just now. We have ICP mass spec, GC mass spec, LC mass spec, and a number of other instruments that are available. And so I'm telling you all this in case students need access to these, letting you know that our NOAA laboratory in Charleston has them. We also have something that's kind of unique. We have a music presence system. So this is enclosed in a greenhouse, and there are 24 tanks. And these are set up to mimic a southeastern salt marsh. So we have an upper tank, so you see in the picture there. Maybe I can point you. Okay. So the upper tanks that you see have spartina grass growing in them. They have sediment in them. And then beneath each one of those is another tank that we call a sump. So by doing some pumping with the system, we can mimic the tide. So we can pump the water up to a high tide mark and then kick the pump off, and then gravity falls back down to a low tide mark. And so we don't move it through time like real tides do, and we don't have magnitude changes, but we are mimicking the flux. So that allows some control, but also getting at a more realistic exposure. And we do have ambient lighting and temperature. So again, that helps with some of the, the chemical breakdown and things like that we want to be able to look at. So we can put multiple species in here. We can do chemical fate and effects testing in there. We can test new technologies in there. We can put in water samplers that haven't been fully validated in the field and use known chemicals to try to work out how well they collect and measure. So it's ecosystem level testing. So after we've established levels in the laboratory that we think are harmful, then we move to something like this to be able to predict better what might actually happen out in the real world. In the field, we can also compare stressor effects. We can take animals from a control site or from the laboratory and put them out in the field at different sites. So deploy them in enclosures and be able to see how they do and measure the chemicals there and get at some of that real-time influence. We do monitoring, like I said. We do water quality assessments as part of all of these. We have the capabilities for sample collection in a number of different environments. And we do have data swarms. We've tried through small bits. Again, I'm telling you this because as students, you might be interested in coming to our lab, and these are some capabilities that we have. Okay, so how do we do this type of work? So you have to look at, what's available in the literature in terms of standard methods. The reason you want to do that is because then your data are comparable and can be used by the EPA, for example, for regulatory. So we're providing the information. They make the regulations on what safe levels of chemicals would be, right? So often we will use standard methods to do that. The species that we pick, we do have sort of a standard suite of fish and vertebrates that are common to our area in Charleston. And that are found along the East Coast and in the Gulf of Mexico. But we're not limited to those. We do stick pretty near shore because that's where the greatest anthropogenic inputs are coming through runoff and outfalls. So if we do work with offshore species, we'll probably work with their larval forms that are going to be having a nursery area up in the estuaries. And then we represent a bunch of different trophic levels, habitat types, feeding types, because different chemicals are going to affect those types of organisms differently. It helps if they are ecologically and or economically important species. This is how you convey your information out there and its importance. If it's affecting, the fish, that's an easy one. If it's a polychaete or something, you've got to make that link that this is the food resource for those commercially, recreationally important fish species. And so the chemicals, we do a variety of things, but we're targeting like where the data are needed. Where do they not have marine and estuary species test data for a given chemical? What are some of the pesticides that need to be re-registered as they routinely have to be done? What is missing maybe from the database for those? And then what are the trends in manufacturing, use, and disposal of chemicals? That helps drive our research as well, what to pick to study. And so it's a tiered approach. So taking that lab, mesoplasm field approach, starting with single species, growing it up to multiple species, multi-stressors, getting a holistic approach to the testing. And it's hypothesis driven, and like I said, we're trying to get those actual threshold values that can then be applied to regulate chemicals. This is us. This is the Ecotoxicology branch in Charleston. And so we have a team of chemists, we have biologists, we have people that are really good at field research, we have people that are good at statistics. And so this team really interacts well to achieve these. And we love to work with students as well. So this is some of the work that we do with students. So the EPP MSI, the CCME students, some of the current ones are from the universities that you see listed there. We also host the Pauling Scholars, the undergrads. We work with our local universities. So we have College of Charleston graduate students. We have folks from the University of South Carolina School of Public Health. And we have NSF summer programs in the area, and they often come as well. One of the things about where we are in Charleston is located on a marine science consortium. So the area where we are is home to not just the NOAA folks, but we also have some folks from NIMS present. We have NIST staff, College of Charleston, the Medical University of South Carolina, and the South Carolina Department of Natural Resources. So it's a great place for collaborative interactions. If we don't have the area of expertise with the instrumentation, maybe one of those other groups does, and it's really good for gaining greater access and greater knowledge power across a whole bunch of different groups. So, we also are very interested in starting at the lower level, so we'll go out to the schools, the K through 12, and do their career fairs and their science fairs and try to get them excited about marine research. And we have a new thing called Charleston Planet Stewards, which is a spin-off of the NOAA Planet Stewards program, where we're trying to gain basically some recognition in our very local area so that people know there's a NOAA lab down the street, that there are scientists there. And so we're going out to particularly Title I schools and being able to show them things like, let's build a floating marsh within your retention pond and try to have the vegetation there, mitigate some of the water quality effects, things like that. Okay, so all of the research that we do is applied. So, we're not the ones that are going to go out and characterize a new species or, figure out why bioluminescence. We're trying to figure out how to take data that can be used for management. So some of our current projects that I'll go over, we have some regional assessments. So regional assessments are, there's an area, for some reason it needs information on water quality, the chemicals that are there, how biota are doing, right? So those type of assessments. So we'll go out and measure the chemicals in sediments. We'll use some tissues as well, like oysters or fish, see what's in there. And then benthic community composition is a really good measure and indicator of the overall health of an estuarine ecosystem. We have a fairly good grasp on what are considered healthy benthic community species versus not. So you can look at that as an index. And then at the same time, take some of that sediment from that site and expose test animals to it and be able to see if there's toxicity. And so putting those three things together is the sediment quality triad approach. And it's a way to really assess the health of coastal communities. We also participate in Mussel Watch. And so that's a NOAA program you might have heard of. This is the longest running monitoring study of coastal and Great Lakes systems, where they collect sediment and they collect mussels. So in the Great Lakes, it'd be the zebra mussels. In South Carolina, the oysters, so depending on what is available there. And they measure the tissues, they measure the sediment for a large suite of chemical contaminants. And they do this, year after year in different places. And so there's a database of information. This helps states and other research partners. It can help students looking for data on what are levels of complex studying out in the environment. You can go to that and look and see if it's been measured there. And it's basically a baseline and then a measure for change. So if there's something that happens at a site, for example, the Deepwater Horizon oil spill, you can then pull up the Mussel Watch data and see, well, what were the levels like before this happened? And that's the only way you really know, what kind of impact is happening. So we've done things like that at the Cape Lookout National Seashore recently. They were really concerned because they've had flooding from recent hurricanes. They didn't have a baseline of what the conditions were like there. They know this is going to happen again. They've got all kind of agriculture upstream and urban things that are going to flood water's going to come down into their national seashore. So we provided the baseline information. Now they're able to use that in future assessments. And then the South Carolina Estuarine Coastal Assessment Program, that's our local South Carolina DNR program, and we've been doing chemistry for them for 30 years. So it's a long-term monitoring project. So we're also doing a lot with our Office of Response and Restoration as it relates to oil spills. We've been studying various aspects of that. The Louisiana sweet crude oil that was spilled at Deepwater Horizon, with and without different dispersants, with and without different shoreline cleaners, so the products that you use to try to mitigate an oil spill, and things that might be tools for cleaning it up, like bioremediation products. Testing a novel one that is iron nanomaterials. The idea is the oil's going to stick to that because they have a large surface area volume ratio, and you're going to be able to take that oil up and potentially pull it back off. So looking at that in these NCCOS systems, being able to help guide the future of response and restoration that way. We also do tool development and technology. So we built these field deployed mesocosms because OR&R wanted to have something that could tote out to the field when there's a spill. So made these enclosures and designed and tested those so that you can put fish and shrimp in there to go to a clean site, collect them, or bring it from a lab, or order them from a biosupply company. Go out to that spill site, put them there, and in real time see what happens. Replenish and see how long the effect lasts and things like that. So that will help with their damage assessments as well. And then, like I said, technology development can be tested in these mesocosms and then also brought out to the field and make comparisons. So in the mesocosms, we can apply a known dose of chemicals, right? So I know how much, of a pyrethroid insecticide I put in there. If I use these samplers and compare them, then I can see what's most effective at measuring that. Then we can move back to the field. And so we're trying some different things because just taking a water sample is not always the best approach. That's a real snapshot, if you will. That's why it's called a graph sample. Versus some things that are out there longer, passive samplers that are taking it in over time. And so we compared a couple different passive samplers, those wristbands that you see up there that were in your backpack. Silicone is a very good material for absorbing organic contaminants, and they're very cheap, and they're not likely to, cost much when they get, lost to tides or disturbances or whatever. So we're using those, and they're working out really well. So the silicone bands were compared, a traditional passive sampler, the PFOS, which is a polyurethane sheet, and then a new active sampler, which is that little ball. And that actually pumps the water through. So then you're getting, so it's accuracy, you're getting a known concentration because you can do volume per minute. So we looked at all of this in the mesocosms, and now we're working with Charleston Waterkeeper, our local efforts to protect the water quality in our tidal creeks, putting them out at some of those sites, and then being able to compare those uptake concentrations. So we're establishing rate constants for the ones that are passive, so we can convert those to actual concentrations, and altogether being able to better predict the chemicals that are in the environment. So we're also working with PFOS. This is a new area of research. There's not a lot of marine and estuarine species data for it. So we're testing with various fish and vertebrate species, establishing acute and chronic thresholds for that. And then we've also partnered with the Department of Defense. PFOS is used in firefighting foams. They're trying to phase those out. They have to come up with new materials. So we're testing 7 of the alternatives to those aqueous firefighting foams and establishing thresholds for those. We'll be able to rank the toxicity of those and help them make decisions on what to go to when they move off of the PFOS compounds. And so that's going to be hopefully supporting the EPA and the DoD as they try to regulate this. I should mention those larval forms that you see there are what we try to focus on because usually the early life stages are going to be your most sensitive to chemicals. PhotoOxidation of Floating Oil is another project. After Deepwater Horizon, it really wasn't known, how much the large amount of UV light that you have in the Gulf of Mexico, how much that's interfering with the toxicity, the weathering, what products are being formed as a result of that. So this is more of a chemistry study. looking at the compositional changes of oil as we apply different things like temperature, UV light, wave action. So we're measuring the changes over time with that, again, to support that response and restoration. And then I mentioned earlier on that climate factors really are going to drive how chemicals move. They're going to drive where they're found. They're going to drive where the different organisms encounter them. And they can actually physically change the toxicity of the compounds. So we're looking at temperature, salinity, pH, which is particularly important for shellfish because of the acidification interfering with their shell growth, and dissolved oxygen. And we're doing that with a number of different species to try to characterize these effects. We did it with oil, we're doing it with PFOS, and again, it's adding to that database of information so that people can understand what is expected from out in the environment. Okay. And then this is the project that we currently have going on in the mesocosms. It's the oil marsh restoration. One other thing that they do when there's an oil spill and it gets up on the marsh is that they try to restore that marsh. You have some options. You could do nothing. You could replant within it. You could replant with native vegetation that you collect from another site. You could replant with nursery plants that you order. You could leave the roots bare. You could put them out in plugs. It's not really known what is the best strategy to regrow that marsh quickly. So that's what we're testing in the mesocosms right now with all of those different treatments. And we're going to see what we would recommend at the end of the day with that. So we'll see how replanting influences that weathering and the degradation. Okay, and so I'd be happy to take questions.
Yeah, we just have a couple of minutes to ask questions. Yes, there's a question.
With the oil work you're doing, are you seeing in the mesocosms any relationship between the presence of the hydrocarbons and the increase in shell disease, or what we call cigarette burn disease in the Gulf? Same thing, it's a bacterial infection in the shell. Have you seen any correlation between that?
So the shelled animals that we have had in there have been juvenile.
Okay.
And so I don't think
They're dying before.
I don't think they're at the right stage to look at that.
Okay.
So that might be better done in a laboratory where we put adult oysters or adult clams in. And
Well, I mean more like decoupage, crabs, shrimps, lobster, stuff like
okay, yeah. So we generally don't put crabs in those mesocosms. We've tried fiddler crabs.
Yeah.
They drown.
Yes.
Because
They need land.
once they have a marsh, but they can't get back up on that platform.
Okay.
So it hasn't worked out very well.
Okay. I know that.
We take out the little crabs to destroy it.
Yes, they do. Cool.
All right, so we don't have any more time, so if you need to talk to Dr. DeLorenzo later, will you be around?
This is